It was during these early days of the project that a pattern of international collaboration was established. The United States was directing the effort and carrying out more than half the work. The rest would be done mainly in the United Kingdom, France, Germany, and Japan. Despite a long tradition in genetics and molecular biology, the UK's Medical Research Council was only a minor contributor. Like the whole of British science, it was suffering from Mrs. Thatcher's myopically stingy funding policies. Fortunately, the Wellcome Trust, a private biomedical charity, came to the rescue. In 1992, it established a purpose-built sequencing facility outside Cambridge, the Sanger Center, named, as we have seen, for Fred Sanger. In managing the international effort, I decided to assign distinct parts of the genome to different nations. In this way, I figured a participating nation would feel that it was invested in something concrete, say a particular chromosome arm, rather than laboring on a nameless collection of anonymous clones. The Japanese effort, for example, focused largely on chromosome 21. Sad to say, in the rush to finish, this tidy order broke down, and it proved to be not so easy after all to superimpose the genome map on a map of the world. From the start, I was certain that the Human Genome Project could not be accomplished through a large number of small efforts, a combination of many contributing labs. The logistics would be hopelessly messy, and the benefits of scale and automation would be lost. Early on, therefore, genome mapping centers were established at Washington University in St. Louis, Stanford, and UCSF in California, the University of Michigan at Ann Arbor, MIT in Cambridge, and Baylor College of Medicine in Houston. The DOE's operations, first centered at their Los Alamos and Livermore National Laboratories, in time came to be centralized in Walnut Creek, California. The next order of business was to investigate and develop alternative sequencing technologies with a view to reducing overall cost to about 50 cents a base pair. Several pilot projects were launched. Ironically, the method that eventually paid off, fluorescent dye-based automated sequencing, did not fare especially well during this phase. In retrospect, the pilot automated machine effort should have been carried out by Craig Venter, an NIH staff researcher, who had already proved adept at getting the most out of the procedure. He had applied to do it, but Lee Hood, as the technology's original developer, was preferred. This early rebuff of Venter was to have repercussions later. In the end, the HGP did not involve the wholesale invention of new methods of analyzing DNA. Rather, it was the improvement and automation of familiar methods that ultimately enabled a progressive scaling up from hundreds to thousands, and then to millions of base pairs of sequence. Critical to the project, however, was a revolutionary technique for generating large quantities of particular DNA segments. You need large quantities of a given segment, or gene, if you're going to sequence it. Until the mid-'80s, amplifying a particular DNA region depended on the Cohen-Boyer method of molecular cloning. You would cut out your piece of DNA, insert it into a plasmid, and then insert the modified plasmid into a bacterial cell. The cell would then replicate, duplicating each time your inserted DNA segment. Once sufficient bacterial growth had occurred, you would purify your DNA segment out from the total mass of DNA in the bacterial population. This procedure, though refined since Boyer and Cohen's original experiments, was still cumbersome and time-consuming. The development of the polymerase chain reaction, PCR, was therefore a great leap forward. It achieves the same goal, selective amplification of your piece of DNA, within a couple of hours, and without any need to mess around with bacteria. PCR was invented by Kary Mullis, then an employee of Cedus Corporation. By his own account, The revelation came to me one Friday night in April 1983 as I gripped the steering wheel of my car and snaked along a moonlit mountain road into Northern California's redwood country. It is remarkable that he should have been inspired in the face of such peril. Not that the roads in Northern California are particularly treacherous, but as a friend who once saw the daredevil Mullis in Aspen skiing down the center of an icy road through speeding two-way traffic, explained to the New York Times, Mullis had a vision that he would die by crashing his head against a redwood tree. Hence, he is fearless wherever there are no redwoods. Mullis received the Nobel Prize in Chemistry for his invention in 1993 and has since become ever more eccentric. His advocacy of the revisionist theory that AIDS is not caused by HIV has damaged both his credibility and public health efforts. PCR is an exquisitely simple process. By chemical methods, we synthesize two primers, short stretches of single-stranded DNA, usually about 20 base pairs in length, that correspond in sequence to regions flanking the piece of DNA we are interested in. These primers bracket our gene. We add the primers to our template DNA, which has been extracted from a sample of tissue. The template effectively consists of the entire genome, and the goal is to massively enrich our sample for the target region. When DNA is heated up to 95 degrees centigrade, the two strands come apart. This allows each primer to bond to the 20 base pair stretches of template whose sequences are complementary to the primers. We have thus formed two small 20 base pair islands of double-stranded DNA along the single strands of the template DNA. DNA polymerase, the enzyme that copies DNA by incorporating new base pairs in complementary positions along a DNA strand, will only start at a site where the DNA is already double-stranded. DNA polymerase therefore starts its work at the double-stranded island made by the union of the primer and the complementary template region.